Hi, I'm Prasad Naidu. Today we are discussing about the blotting techniques. So, this is visualization of specific DNA and RNA protein among the many thousands of contamination molecules requires the coverage of numbers of techniques which is collected by the term as a blot transfers. So, this blot transfer we're talking about the blotting techniques. This blotting, first of all, these blotting techniques here is DNA, RNA, and proteins. First of all, DNA is we can detect the southern by using of southern blotting technique, and to detect the RNA by using of northern blotting technique, to detect the protein by using the western blotting technique. So these are the three components: southern blotting technique and northern blotting technique and western blotting technique. Coming to the southern blotting technique, in 1975, Edward Southern is developed on the technique that widely used to detect the fragments of DNA. This requires of separations of DNA and DNA fragments by agarose gel electrophoresis, and DNA fragments are blotted onto a strip of nucleocellulose or a nylon membrane. Identification by hybridization with the label and complementary nucleic acid probe. Southern blot is a method transfer from the DNA from an agarose gel nucleocellulose filters on which the DNA can be detected by using of suitable probe like a complementary DNA or RNA. The DNA sample is digested by restrictions like endonuclease. This is we are talking about the procedure. The DNA sample is digested by the restriction endonucleases, producing the small fragments that are amenable for analysis. Fragments are separated by agarose gel electrophoresis, that is agarose gel electrophoresis or polypeptide gel electrophoresis. The mobility of nucleic acids in the agarose gel is influenced by the agarose concentration, molecular size, and molecular conformation of the nucleic acid. Then agarose concentration of 0.3 to 2% are most effective for nucleic acid separation. Like proteins like nucleic acids migrate a rate that is inversely proportional to the logarithm of molecular weight. So separated nucleic acids are visualized fluorescent dye, ethidium bromide. The agarose gel is soaked in the solutions of dye and washed for remaining excess dye. The illumination of the rinsed slab with the ultraviolet light reveal red-orange stains where nucleic acids are located. So ethidium bromide strains both single and double strand nucleic acids. The fluorescence is much greater with the double stranded DNA molecules. The electrophoresis can be preferred with the dye incorporated in the gel and buffer. This has the advantages that gel can be illuminated with the ultraviolet light during the electrophoresis to view the extent of separations. The mobility DNA is reduced to 10 to 15% in the presence of ethidium bromide. This ethidium bromide must used with the great care. It is potent mutagen. Gloves should be worn at all the times by using the washed, like using dye solutions and handling gels. Some newer fluorescent dyes are produced. Molecular probes offers the several advantages: less toxic or 5 times more than sensitivity ethidium bromides. The labeled DNA radioisotopic cytochrome P32 and 5' to 3' ends. This cytochrome that probe 32 is a strong beta emitter. The bonds of labeled the DNA on the electrophoresis gel can be located by the autoradiography. That labeling molecules before the analysis coenzymes biotin formation strong complex with the enzymes linked to streptovidin. The polyacrylamide gel electrophoresis is useful for the small fragments of DNA up to 350,000 daltons in molecular size. The large molecules DNA could be separated by pulse field gel electrophoresis, that is PFG. Blotting techniques like here we're talking about the blotting. Transfer the DNA from the gel to nitrocellulose membrane done. So weak acid treatment to depurament fragments the DNA. Those make it smaller or easier to elude the formation of gel. followed by denaturation with the strong bases, neutralization, hydrolyzation of phosphodiester backbone at the depurinized sites. Single strand bind to the membranes or efficiently. A buffer is used to facilitate the transfer. So original methods, the transfers are realized capillary actions and vacuums or pressure systems can be used to the speed the transfers and foster the most transfer to offer for like the uses of electro blotter, and electro blotting is a process usually completed is 1 hour to 4 hours. Hybridization assays, all hybridization assays are based on the ability of nucleic acids from the specific double strand hybrids. The process requires a probe that can target nucleic acids, allow the specific complementary base pairing. The method is detecting any resulting to double strand nucleic acids. So conditions are high strategies here, hybridization assays, low salt concentration and high formaldehyde levels and high temperature. As the stringency of the assay is lowered, increased number of bases mismatches are tolerated. Conditions of high stringency is required exact base pairing. So sometimes that time is required to hybridize the probe to the given fraction of target remains proportional to the probe concentration. The rate of hybridization reactions is influenced by the temperature of ionic strength. Above the temperature, no stable hybrids and present with the divalent cations like magnesium have stronger effect on hybridization. The unbound probes are removed by the washing, and probe bound to the membrane is detected autoradiography, which reveals the DNA fragments to which the probe hybridized. So finally, we'll come to the application. Southern blots are used from is gene discovery and gene mapping and evaluation of development of studies and diagnostic forensics and deltons and insertion and points mutations and polymorphism, structural rearrangements, and allow for the determination of molecular weight and restriction fragments and presence of particularly bites with the DNA in the sample. These are the applications. So coming to the northern blot, southern blot is for DNA, northern blotting for RNA. This northern blotting technique is a way detection of specific RNA sequence developed from the James Alvin George stack. RNA molecules have defined length and much shorter, and the genomic DNA not necessary to be cleave the RNA before electrophoresis. RNA is more susceptible to degrade the DNA. RNA sample are separated based on the size of gel electrophoresis, and also here we will discuss about the RNA. This RNA is blotted onto nylon positively charged membrane. The membrane is placed an hybridization buffer with labeled probe, usually in DNA. Labeled probe is detected in autoradiography. Expressions of pattern sequence interest in the different samples can be compared here. Applications, these applications we all know, these applications, the standards directly gene expression at the level of mRNA, messenger RNA, detection of mRNA transcript transcript size. The study of RNA splicing can be detected here. Alternatively, spliced transcript, and the study of RNA is half-life. Some disadvantages here. The time consumption procedure and RNA samples can be degraded by the RNases, and usually radioactivity probes that detect the multiple probes in the problem here. Western blotting, we all know southern blotting for DNA, northern blotting is for RNA, and western blotting. This western blotting is an immunoblotting technique, rarely specified binding between the molecules and interest probe to allow the detection of molecules interest in a mixture of many other similar molecules. Western blotting technique, molecules interested with the protein. The probe is typically an antibody raised against the particular proteins. This SGS plays polyacromate gel electrophoresis technique is used to pursue for the Western blotting techniques. Proteins is sampling subjected to electrophoresis SGS polyacromate gel. So electroblotting technique transfers the separated proteins from the gel to the surface and nutracellular membranes. So this blot is incubated with the generic protein such as milk, which binds to the remaining strict places in the nitrocellular wrapper. This antibody is specific protein to interest with the primary antibody added to the nitrocellular wrapper sheet and react with the antigen, and only the bound that band, some band here is containing the protein interest with the binds antibody, forming a layer of antibody molecules. So following several rinses of removal of non-specific bound antibody, the antibody antigen complex like nitrocellular sheets, and incubated with the second antibody, and which is specified recognized the domain like specific recognized with the like primary antibodies like antibody 2 react with the labeled and covalently liked with the reporter like enzymes visualize the protein antibody 1 and antibody 2 complexes. Finally, we will be talking about the applications of Western blotting techniques. The confirmatory HIV tests and employment is like major here. We will test of Western blotting technique is used for the Western blotting technique is HIV to detect the human antigen human samples. So proteins are known as HIV influenced cells separated blotting techniques a membrane, then serum to be tested applied like in the primary antibody incubations. Free antibody washed away this like here signaling added like what finally our assumption is we're talking about the HIV antibody detecting here we using by using of Western blotting technique. This Western blotting technique here is protein we are detecting here antibodies. So finally we're talking about the blotting techniques is Southern blotting technique, Western blotting technique, and Northern blotting technique. Southern blotting for DNA identification. Northern blotting technique is RNA to detect the RNA, and Western blotting technique is to detect the protein. So finally, these are the blotting technique. Thank you.